Hi, thank you for joining us today at the Precision Medicine Leadership Summit. My name is Hannah Mamuszka. I'm CEO and founder of Alma10, and I'm joined today by Satabhisa Mukhopadhyay, who is co-founder and chief scientific officer of 4DPath, an up-and-coming digital pathology company that has the potential to revolutionize oncology care by digitizing biomarkers that we typically think of as requiring IHC, PCR, and NGS. And today we're going to talk about what the potential is for digital pathology to revolutionize how cancer patients are treated and managed, and how we can expedite and democratize care. So first, we'll start with some basics. Satabhisa, tell me a little bit about your background and about how you got started at 4DPath.
Hi, Anna. Good to be here. Thank you for having me. So I am, by training, a theoretical physicist, did my master's from Cambridge University and PhD in high-energy physics and string theory from Rutgers. But I naturally gravitated to cancer cell biology, systems pharmacology, genomics, all this sort of stuff, and worked for about a decade in MIT and Harvard Medical School until we founded 4DPath with some very exciting people I know. And always the emphasis had been on finding more data, more hidden data. There are a lot of hidden data in the systems we use. And how can I capitalize on that? And at 4D, we have an exciting system that can read way more data starting from just the H&E digital biopsy image, and can extract the translational information, diagnostic information. And we have started this company from 2016. We have US patented technology. We're going to regulatory space, and a lot of exciting things are coming up.
That's amazing. So what you're saying is that you're doing all of this from just H&E. I'm not a pathologist by training, but I am a laboratorian, and I've spent a lot of time around pathology. And typically, I think most of us think, maybe not accurately, that H&E is kind of the first step that we do to confirm diagnosis and to take a first look at the cells, but it's really not the confirmatory piece of information. It's not really where we get our biomarker information from. So are you saying that we should be thinking about H&E differently?
Yeah, absolutely. So H&E, as you said, is the very first step. It's a very stable stain. So you can color the nuclei and the cytoplasm, the rest of the cell differently. So pathologists can easily see different features that are useful for diagnosis. Then people don't look at that other than if they're using some molecular stain, and also extracting samples to send to sequencing, NGS, all that stuff. But what we are saying, inside the same H&E, there are signatures of cell-to-cell talking, and we can extract that from H&E itself. So without the aid of any other molecular stain, any sequencing or NGS panel, anything, we can extract tumor molecular information, and thereby can confer various diagnostic and prognostic decisions that are useful in translational medicine.
So the name of the company is 4DPath.
Yeah.
Is this really about AI, or is this really about machine learning or deep learning? Can you describe a little bit about what each of those are and what's really most applicable to how we should be thinking about 4DPath?
Yeah, it's a great question. So first, let me tell just in very simple term what is AI, ML, and DL. We talk a lot about it these days. So AI is the broad discipline of the wish to build a system that is as intelligent as a human, in the sense that under a given circumstance or situation, it can reason like human or respond like human, and thereby behave like humans. So that's a broad field. But within that field, ML or machine learning is building the system of algorithms that can learn from data or the experience of data and build a hypothesis from this experience, then validate this hypothesis on another set of data from which it did not learn the underlying model or hypothesis, and then iteratively correct this hypothesis by penalizing the system. Now within ML, there is a set which is called artificial neural nets, which are like a model is built based on, like, motivated by the neuronal nets we have in our brain in a very crude way. But then deep learning is the methodology where, by using various handcrafted special architecture of this neural net, you can mine a very large set of data, and with efficient timing, with efficient speed, and thereby you can do decisions, try to do decisions that require very large data. So that's what it is about. Now, if you think of 4D path, 4D path is an automated system, some kind of intelligence that is predicting the hidden data, the hidden cell talking from HNE10. But we are not AI, ML, or DL in the sense that we don't learn from data. We're not learning from the experience of the data. Inside our prediction core, we have a priori hard-coded reasoning that is coming from the underlying tumor biology. We have a lot of knowledge of the fundamental processes in tumor biology. We hard-code that, and then we validate the hypothesis. That's what 4D path is about. And cell-to-cell talking is another dimension. So intrinsic time. So it's 4D. We have 3-dimensional flesh, the tissue, and then intrinsic time. That's about 4D.
That's fascinating. So what you're saying is that you can do the aspects of pathology in terms of diagnosis, staging, and then you can also do the aspects of biomarkers to confirm presence or absence of particular markers that we might usually think would require either an antibody stain, a PCR reaction, or an NGS run. But then when you talk about the cell-to-cell communication, that sounds like a whole other dimension of biomarker discovery. You know, and when you look at the existing literature and all of the data around targeted therapies, you know, there's always sort of a question of why are the response rates not maybe higher for some of the targeted therapies? And do you think that some of this cell-to-cell interaction and some of this crosstalk could maybe be better optimized to determine who's more likely to respond to therapy?
Yes, of course. Yes, of course. So, for example, you see that if you talk about the PARP inhibitor therapies for BRCA mutation or people harboring HRD, so then it's like a whole umbrella of instabilities, right? You might go one by one by measuring different mutations, but then it's a whole umbrella of instability that you can tease out from cell-to-cell talking, and thereby you can specify the degree of efficacy the PARP inhibitor will have. Same story for example, the CDK4/6 inhibitors. So these are like, I would say there is a huge signature of this competition between the tumor suppressor and oncogenes and the addictive nature of cancer, so that you can better predict who is sensitized to which targeted therapy. All you can do much before NGS panels or IC10, right at the time when the biopsy image is digitized and uploaded to our system.
So how long does all of that take though? Because I mean, right now, if you think about sort of the standard process of a patient or a pathologist or an oncologist, you have a suspicion of cancer, you have a biopsy that's taken. That biopsy, first thing, is sent to H&E, and then based on the H&E results is going to be sent for further staining or further biomarkers. How long, and that process, you know, can take, you know, a week or 2 weeks, or if you're doing a full NGS panel, maybe it takes a month. How long are you envisioning that it's going to take to get the result back to the physician and patient?
Oh, we are practically real time. The reason I'm saying that, we are under a minute effectively from biopsy. Yes, I mean, I can tell you for us, a batch of typical 200 whole slides, whole biopsy slide would be like, on an average, it's like 30 seconds. So, and the sizes vary. So you can imagine we are under a minute, and we have a capacity, we are building capacity to scale up. So depending on all these processes, like you still have to digitize the image. That part we don't have control. So I mean, it should be within a day. People, whenever they upload, they will get in almost in real time. So I guess patient would get in a day or yeah, something like that.
Wow, that's amazing. And I mean, the ability to use a digital solution like that would mean that a patient wouldn't need to be seen at a center that had a full complement of PCR machines or NGS machines, or wasn't willing to send out to a outside lab. I mean, that would tremendously democratize the ability of patients to get appropriate biomarker testing.
Yeah, that's true.
So, given the potential scope of that, how do pathologists feel about the data that you're developing and what you're doing? And do you think that there will be any major barriers to getting pathologists on board to accept your technology?
Well, so we are not replacing pathologists. We want to give them a automated parallel system, an alarm system that can prioritize their workload and then can give them flags and checks about subtleties and then complexities. There are a lot to talk about, subtleties, complexities that they face, and then that can reduce work volume, but also faster turnaround time, low cost, and a second adjunct opinion, which doesn't need their time. It's fully automated. So people like that idea, and we provide, to facilitate that, we provide a lot of parallel internal consistency check that are quick for pathology, so that they can easily use and prioritize. But moreover, we have overlap or alignment with patient data, so that's another satisfaction. For example, here I can quickly mention, we are going over some patient data and pathologist data, and you know that you have biopsy sample, but then later many of them will have resection. And resection is the full tumor, has the full signature, the true signature of what the tumor has. And then we discovered that 20 to 25% of biopsy grading by pathologists are wrong with respect to resection. And that's because biopsy can have undersampling, or because of stain variability. It's you do grading by Nottingham prognostic score, where you just look at certain features of the tumor and then score them together, pack the score together, and that's the way you do. And some of these features you may read wrongly if there is stain variability. So it's natural, there are mistakes, but we provide that these alignments, and then where the pathologists are more prone to make mistakes, this kind of thing that facilitate. But we don't load any of their time. They don't have to go and check with our tool interactively, or don't have to pre-annotate or anything. It's a parallel system that helps you. So we got good response, in other words.
What does your data generation process look like? I mean, I would imagine that there are a lot of data banks of H&E. I don't know how many are digitized, but what is your data validation process been in order to give pathologists confidence in your platform and in the tool that you're developing?
Yeah, that's a great question. One thing is important here, that you work close to this real life hospital data directly. You work on that, because in real life hospital data, there are lots of nuances. And for example, for a case about big tumor, you can get many slides, but you have to give verdict for all the slides. You have to take into account of heterogeneity. So we work on very high quality data. We have a very good relationship with Leeds University Center, and then we have capacity of getting. We have an agreement with them, getting large volume of data, including patient data and ethics clearance. So we have a very good arrangement. And then we are also working with other labs and hospitals. And so we work on real clinical data with all nuances. And that gives us very much confidence, and pathologists also like that.
Well, and then what about the other hurdle, shall we say, being the regulators? How does the FDA think about digital pathology? what are their perspectives on risk? And you know, if you're talking to the FDA, how is that going?
Yeah, so that's a great question. Yeah, that's a serious hurdle, because for AI and digital diagnostics, what are the sources of risk, how you are quantifying risk, how you're mitigating risks. Now, for other AI systems, it's a black box, so it's really hard to quantify all these steps, and that's why the AI diagnostic systems maybe eventually, uh, at least for now, are limited to very narrow range and standard of care, a very special condition and all that. And but for us, we have a different story, because we are a white box, so we can just dissect where are the risks that can come from, and then how do we quantify what are the orthogonal or independent tests or gold standard or alignments we should show to check that this risk is in control. So we do that consistently. Of course, FDA is the best authority to talk to get to know all of these questions, and we're doing that. And I think it's a good experience. And another important thing is that how much you can align with patient outcome and outcome data is the ultimate gold standard. For example, for HER2, you have a lot of variability from the antibodies if you do IHC, and there is a broad range standard of care from local lab to central lab, from very experienced pathologists to average pathologists. And then FISH is maybe more accurate, but neoadjuvant therapy response for herceptin treatment is even more accurate, right? Because eventually we are doing this diagnosis for some selecting treatment and giving the treatment quickly that will be effective. So all these things are in play, and I think we are in good shape to do them one by one.
Mm-hmm. You know, I'm thinking about, you know, what are the barriers maybe for you to access the market? As far as digital scanners go, how broadly are digital scanners currently used in pathology? Is that a standard activity, or is that something that's sort of specialized?
Like in Europe, and particularly in the UK, because it is mandated by NHS, it's standard practice. But in US, I think scanners and digital images are being used for research purposes mainly. It has not come to pathology diagnosis yet, but I think we have the hope that with this regulatory clearance and more discussing about, with the ability of more and more discussing the risk quantification and management, I think things might change, and things should change, because it's very hard to like scale up. I mean, with digital solution, you can scale up. But there are all sorts of applications here. I hope that there'll be, as I was saying, that central labs, local labs, say hospitals in remote setting, there can be many niche where it can start, more use can start of digital scanners and digital pathology. And so we are exploring that.
Yeah, that's fascinating. Are there a lot of, do you haven't seen any variability between different scanners? I know there are a few different brands, or is that something you've controlled for?
So yeah, so that's a good question, and I should mention here that there is a lot of variability in digital image, which is one of the obstacles. The variability can come from staining, slide preparation from that side to even scanning, and then to the digital packaging across the platform. So first of all, we don't work on colors, so we cut down all these variabilities to a lot of extent, so that we can even simple pre-processing computer vision algorithms. We have certain proprietary algorithms that can take care of this much reduced noise. And then, so that's very good. And the way we are built, we are platform agnostic. So we have no problem. We can access many kind of clients, whoever is working with what format. We are format agnostic. And but then there are other kind of noises, that is patient to patient variability, right? So, and that noise is useful for clinical treatments or guessing the targeted therapy, but the way we read from underlying fundamental biology, we can still keep that useful noise and use that, whereas we don't have to see the variability from the staining or scanning or different formats. So it's all good news.
I don't think I've ever heard anyone in diagnostics describe useful noise before. That's fantastic. What do you think that the most immediate term applications for your platform are in oncology? I assume oncology, but within oncology.
Yeah, within oncology, I think the immediate use is screening patient population very easily, right after the biopsy is digitized and uploaded to a system for really targeted therapies, like we see currently for PARP inhibitors. We are doing interesting work, and we see like really exciting data. And then CDK4/6 inhibitor, and also for lung cancer panel, ALK and other rearrangement, and then pan-TRK inhibitor type of thing. So these are like really very immediate signatures that we can just read from biopsy, and that can help pharmaceuticals as well as in oncology to screen patients, even before for new drugs, and even like screen for trial.
Yeah, I mean, if you look at the trends in oncology lately, a lot of the pharma companies are really trending towards sort of these micro groups that are no longer defined by pathology, but that are really defined by a genetic aberration, a fusion, or some other mutation. But finding those patients is a challenge, especially if it's dependent on NGS. And so, I mean, if you could really talk about moving that to a digital application, I mean, not only would that be great for pharma, because it would help them find more patients, but the way that would potentially democratize care and spread the ability of getting those incredibly effective therapies out into patients who, may not have the ability to get sequenced by NGS. That would be a revolutionary change in oncology. That's really fascinating. So, well, thank you so much, Satabhisa, for joining me today. And everyone, please stay tuned for question and answer coming up next. Thank you for joining us for some questions with Satabhisa Mukhopadhyay, who is Chief Scientist and co-founder of 40Path. I hope you enjoyed our discussion around the potential for digital pathology. Satabhisa, the first question is, with known heterogeneity in oncology and tumors, particularly in solid tumors being such a challenge for both diagnosis and management, do you think that the 40Path platform is something that can address heterogeneity? And do you have any sampling issues due to heterogeneity of tumors?
Okay, thanks, Anna. Hi. So it's a great question. So we can tackle heterogeneity because for a large tumor, all the slides are prepared. We can take them equally. We scan locally. We use the entire slide, and we can tackle the heterogeneity because we can stratify different local signals. And so we have a systematics to tackle this heterogeneity. And I think with respect to certain mutations, like for example in HER2, we see that when you use our extraction of this heterogeneity, effect of heterogeneity, when you link it to the patient outcome data, you can make a lot more sense. That you know that you are taking out this heterogeneity, and immediately to the neoadjuvant therapy. If you link it, you see that it makes sense. So I think there is a big scope for us to answer questions in heterogeneity. And that's a big thing,
there have been a few other pathology AI companies that have been making some noise in the media lately with either notable investments or notable partnerships with pharma. Can you describe a little bit how your platform is differentiated?
Yeah, absolutely. So the first thing is that by the very nature of how these how the core of the algorithm works, so we are not looking at like cell identification and cell classification. So then, and we are also not working on colored images. We're working on grayscale images, suppressing a lot of variability from sample preparation and all, and scanning variability or the scanning platform variabilities. Those technical variabilities or preparation variabilities we are overcoming. But not only that, AI for a reason, because they are trying to learn from experience of pathologists. That's why they're translating this problem of digital diagnostics into cell identification, cell classification. But then at that abstraction level, there is a lot of cell-to-cell, patient-to-patient variability coming in, which I call good noise otherwise, which you should use for even heterogeneity and all this question. But if that noise disturbs you, then how can you use it? So, but that's why the main difference, I would say, not going to much technique or any complications, that our abstraction level is different. So we, instead of cell identification classification, are going to translate all this morphology into process and distortion in process, and then connect to checkpoint distortion, pathway distortion. Then you can use this noise, stratify this good noise of variability, and doesn't affect your problem. So we are very different in that. And what does that enable us? We can directly read the molecular profile of the tumor, which you would not otherwise do with these AI techniques, because like the pathologists, they don't read those molecular profiles often. There's sometimes some signatures. You can co-associate some morphologies, but you cannot directly read the pathway distortion. We can. So in that respect, you can see that we're very different.
So my understanding of that is, I mean, there's well documented subjective bias within standard pathology. And so my understanding of that is that some digital pathology methods bring that bias into their interpretation. But if your interpretation is entirely separate and you're not basing it on the pathology bias, then you don't sort of build the problem into the new algorithm. Am I framing that correctly?
Yeah, absolutely. Because as you know, that morphology of two similar cells, I mean, two very different cells can have same morphology in different tissue contexts, but also, and then you cannot separate them. There will be a lot of, and pathologists will have that bias. So you're framing it correctly, absolutely.
The next question from the audience is, you've talked mostly about solid tumors, but do you think there's an application for the 40-path platform for liquid tumors, for leukemias and lymphomas?
Oh, absolutely. Our method can work on even primary cell culture. We're working on cytology, cervical cytology, and we plan to go to non-solid tumors.
Oh, interesting. And then I guess the next logical question is, if it works in normal cells, are you thinking about, not working maybe on right now, but are you thinking about other disease applications where pathology is a question?
Yeah, absolutely. Like this methodology is a systematics for reading signature of inflammation, right? But cancer is very addictive system. But other type of inflammation, which may be some kind of benign hyperplasia kind of thing, or other inflammation where pathology could be useful, but maybe the methods have not been standardized, or the difficulty by without a digitized solution would absolutely be able to go there. But it's down the road.
Sure. I mean, you can think of, probably top of my mind, an area of major pathology question is NASH, which is an emerging area where the biopsy, the liver biopsy, is not the best gold standard, but we don't really have anything else. But some of the challenge with that biopsy, you wonder if could be solved by a digital solution like you described.
Yeah, absolutely. And actually some sort of like benign chronic condition might look like some pre-malignant, but you have a huge space there and huge need there to differentiate. So absolutely.
Right. Well, thank you everyone who submitted questions. That's the end of our question and answer period for this session. Please tune into the next link for the next upcoming session. Thanks, Satabhisa.